Hi, my name is Wieland Huttner. I was a group leader at EMBL from 1985 to the end of 1990 in EMBL Heidelberg.
Okay. How did you get to know about the EMBL, and how did you get recruited at the EMBL?
So I guess it was my friend Kai Simons, who recruited me. I was a group leader, a junior group leader at the Max Planck Institute of Psychiatry. That was the name at the time, in Martinsried near Munich. And I had started there in 1981. And I had a 5-year contract, which ran until the end of 1985 with a 1-year extension. But I got to know Kai Simons, and he said, "Oh, what you have done is great." We had worked on a post-translational modification called tyrosine sulfation. And he said, "Why don't you come to EMBL?" Graham Warren had left, and he wanted someone with a similar biochemical knowledge. And that's how I got recruited.
How was EMBL in 1985?
Fantastic. I mean, it was the most international research institution or institute that I had worked until still in my life, that I've worked in my life. And it was fantastic.
In which way the EMBL at the time enabled your science? I mean, what was the thing that, in moving, you found more enabling for your science at the EMBL?
I mean, of course, the cell biology program, which I joined, was at the time the world's best cell biology. And our research on this post-translational modification, which was a modification taking place in the Golgi apparatus, took us more and more and more into the field of membrane traffic. So basically, that was a perfect environment for me at the time.
In thinking back about those 5 years, what was the, scientifically, the thing that you think, left the mark of the work that you have done at the EMBL?
Well, it started right at the beginning. We worked on, as I said, on this protein tyrosine sulfation, and there were certain very widespread secretory proteins called the chromogranins or secretogranins that undergo tyrosine sulfation. And we wanted to clone them, the cDNA clone them, which at the time was not an easy thing. And the problem was that one of the proteins, chromogranin A, that we had cloned in competition with a group in the US, the cDNA coded for a much shorter protein than it ran on a gel. So the aha moment was when I, with the help of Bernhard Dobberstein, who was a neighbor next door, performed coupled transcription translation to prove that what we had cloned ran exactly right in the SDS gel. Oh, and when I presented that at a meeting in the United States, there was I don't know what the English term is. In German, we say a Raunen went through the room when I said, "Oh, we have done coupled transcription translation, and that's how we proved it." And then the competitors said, "Well, we are not so lucky to have a Bernhard Dobberstein next door. I'm sorry."
So I guess this was one of the advantages of the EMBL.
Absolutely, top-notch technology and a very, very friendly atmosphere.
Yeah, and I mean, a lot of people that I have interviewed said that back then it was so easy to interact with basically everybody at every level.
Absolutely, it was simply, I mean, at the time, I think for me, it was the best institute that I have ever worked in at the time. I mean, now, of course, being a founding director of a Max Planck Institute, I think we are equally good and equally international, actually. But we come to this maybe later, but when I was asked to be a founding director of the Max Planck Institute, I went to the president of the Max Planck Society and said, "My dream is to make an East EMBL." And the president said, "Herr Huttner, you can do that, but you cannot call it that. It's a Max Planck Institute."
Okay, that is, yeah, I guess a good answer and in line with the...
So it shows that EMBL left a permanent mark in my career, no question.
Yeah, I mean, it's remarkable because, as you said it yourself, it wasn't a long time, and there's a lot of things that we know you have achieved. And one thing that I want to really talk about is the establishment of the PhD program. Having been a PhD student myself, I remember, how unique and how great the program was, and how dedicated the training and all the opportunities that we had, how special this was. And so I wanted to, understand how this came about. I mean, I think back then it was one of the most forward-looking programs that we had in the world, or in Europe, certainly.
Thank you, but I actually think that's true. So what happened was the following. One day, the director general at the time, Lennart Philipsen, came to my office, tapped me on the shoulder, and said, Wieland, I want you to organize a PhD program. At the time, EMBL had a tiny PhD program, which wasn't worth its name, frankly. There were very few official students. The vast majority of the pre-doc students at EMBL were unofficial. They knocked at the door of a group leader and were hired. So I decided to change that completely, to introduce a central selection, to introduce a thesis advisory committees, to introduce an obligatory pre-doc course, and to transfer all the unofficial pre-docs into the program so that we had no more unofficial pre-docs. They were no longer allowed to have an unofficial pre-doc. You either get a pre-doc through selection or you don't have one.
So did you look at, examples in the US of some of the, what was the genesis?
I mean, so I guess this is long ago, so I hope I remember this correctly, but I think the idea with the thesis advisory committees was given to me by David Meyer at the time, who was a group leader, if I remember correctly, and the rest was obvious. You could not have two classes of pre-docs, official ones and unofficial ones, and you needed a central selection. You didn't need to look into the US. It was just obvious that one needed that. And you needed a pre-doc course so that all the students benefited from the fantastic knowledge that existed at EMBL.
So I can remember also this great sense of being a class of fellows, and I mean this played a great supportive role in the whole operation. I think all the students, they felt like they were taking part in a big endeavor. They were trained all together for this endeavor. This might seem like a compliment only, but really, I strive to organize my own PhD program this way, and it's not simple even nowadays. I think one question is, what enabled to do something like that at EMBL? Was this because there was no bureaucracy, or you could be independent of the bureaucracy?
There was clearly no bureaucracy. That was very good. And well, I mean, Lennart Philipsen, he just said, listen, Wieland, do it. And so I did it. And actually, when I became one of the founding directors of the Max Planck Institute in Dresden, we may come to that later in the interview, I was actually the head of what's called an International Max Planck Research School. It was one of the first PhD programs within the Max Planck Society that our institute set up. And it followed, in essence, the EMBL paradigm.
Yeah. I mean, I think the other thing that was great of having a shared initial training is that you somehow uniform the knowledge of the students that might have very different background. This was something that, again, is somewhat something that was intended, or is somewhat a byproduct of that organization?
No, I think it was intended that you say you want the students to benefit from this incredible knowledge that existed at EMBL. And the thesis advisory committees, as I said, David Meyer gave me an idea there, but I also felt that it was important to make PhD students not be 100% dependent on one supervisor. I had experienced cases, not necessarily in my career, but next door, where there was really where PhD students were not treated right. And so I said, if we have these TAC committees, these thesis advisory committees, that's a way of preventing misuse.
Yeah, no, that's great, because I mean, there's somebody else that is going to have some interest in you that you can refer to. This, I think, is now the standard of a lot of PhD committees. But
well, and when you have the yearly or half 6 monthly meetings, it is, I think, now a standard thing that the first supervisor leaves the room and the other two supervisors ask the student, how are things?
so let's maybe move to what happened after the EMBL when you left the EMBL. So you moved next door, I guess, but you can tell us about that as well.
I had two offers to go to the neuroscience, the newly founded neuroscience center in Hamburg, or to take the succession of Wieland Huttner, the chair of neurobiology in Heidelberg. And the University of Heidelberg made a very, very, very good offer, which almost matched the Hamburg one. And of course, the environment in Heidelberg was much better than in Hamburg. So I decided to not go to Hamburg, but stay in Heidelberg. And I had decided that I would radically change fields. I remember my farewell party where I announced what I would do, what I planned to study, which is to identify the genes that make the human brain big. And everybody thought, this guy has gone totally nuts. He's completely, it was really funny. And frankly, our research at the University of Heidelberg didn't take off very, very fast, because I was also the head of a so-called Sonderforschungsbereich, which is a funding institution, a way of funding research in Germany from the German Research Foundation. And I was so influenced by the EMBL that I changed the institute from a one chair, one empire type structure into a group leader system. We had 8 independent group leaders. My group was one of them. But to support them, I had to basically give a lot of the money that I had been given from the university, the so-called Aversum, to the group leaders. And to keep the whole thing going, it was essential to get funding from the Sonderforschungsbereich. And that took a lot of my time, which meant that the new field, developmental neuroscience, it took a while until it took off. It actually took off with the move to Dresden 10 years later.
Okay, but you were still already trying to,
Yes,
mold the new place to some of the best aspects of the EMBL.
absolutely. Yeah, I mean, I changed it to a group leader system, and my successor has kept that structure. It's now a very successful institute in the University of Heidelberg.
So, and coming back to your, change in scientific topic, was this going through the fact that you have been, studying also neurosecretion, and so you went from the cell biology by the neurons into brain organization, or it was a different?
And so I had been studying via the tyrosine sulfation thing. We came to the chromogranins, which turned out to be great model systems to study the formation of secretory granules as one type of neurosecretory vesicle, and then the synaptic vesicle as the other type, which I had studied already during my postdoc with Paul Greengard. But somehow I was not satisfied with what I was doing. I said, my God, I mean, is this to identify the sorting of a protein to a granule? Is that really it? I mean, is that really the question you want to ask for the rest of your life? And I decided that the increase in evolution of the human brain was a bigger question, and I decided to go for it.
Yeah, I mean, it's very bold, because I as a scientist know how much you tend to be attached to what you're doing and being in the comfort zone, technically, and all the background and everything. So moving, stepping aside and starting something entirely new is very brave. But as you say, I mean, this is like almost a philosophical question in science, how what distinguish humans from primates.
Yes, absolutely. Well, we are also primates, but the good thing was that I introduced a cell biological approach to developmental neuroscience. I think actually that we were the first to really do this cell biological approach in a rigorous way. And we said, let's study the neural stem cells in the developing brain and the progenitor cells from a cell biological point of view, studying their polarity and so on. And that then eventually paid off in the end.
Then you stayed in Heidelberg for a while, and then we know that you met.
Stayed in Heidelberg so at the university. We stayed in Heidelberg from 85 to 2000, 5 years EMBL, and then 10 years university.
Okay.
And then in the middle of there was German unification, the wall had come down, and in the middle of the 90s, I was approached by the Max Planck Society whether would be willing to be involved in founding a new Max Planck Institute in the East. And I said, yes, I am willing to consider that. But I was in the middle of renewing the SFB, the Sonderforschungsbereich, for the last time. So I said, but you must keep this a total secret, otherwise they will kill my Sonderforschungsbereich, and all the groups that are dependent on it will suffer. So I was invited by, I will never forget this, in January 96. I was invited by the committee and I presented my thoughts. I said the two cities that they offered, Halle or Jena, I said, I'm not available. I'm only available for Dresden because that's the city that I think is coming up and coming. And I said, and we also need, we all directors are equal at Max Planck, but we need one director who is really outstanding and the world knows the person and so on. So Janni Nüsslein asked me and said, Herr Huttner, you are thinking of someone. Of whom are you thinking? And I said, I'm thinking of Kai Simons, because Kai had impressed me tremendously as the program coordinator in EMBL. Kai is a fantastic person, as we all know. And so I suggested Kai, and Kai came back to me and said, so Wieland, I hear you suggested my name. What do you think? And I said, Kai, I think this could be a unique opportunity. So then the two of us, and together with Tony Hyman and Marino Sirial, set off this to Dresden to found this new institute, which was modeled after EMBL. And I think it has worked. The four of us were essentially all EMBL virus infected people. And we are friends. And so I guess the one reason why it really worked in Dresden is that there was complete trust amongst the four of us. And we all had the same spirit, this EMBL spirit.
I mean, and coming back to the research you were doing at the EMBL, how do you think it, if you look back to those years, I mean, how do you think these, the impact of that looking, 30 years from that moment, what can you tell us about that?
I mean, so one is that one of my students, who is now world famous, Christoph Niers, who's used to run the IMB in Mainz, had succeeded in purifying the enzyme that catalyzed this post-translational modification tyrosine sulfation. And that was an amazing achievement because this was the purification factor, the foldness of purification, I think was the greatest at the time for any membrane protein. I think he needed to purify it 140,000 fold, if I remember correctly. So that was really absolutely stunning. One highlight, and the other highlight is fair to say, I think that what we had found on Sharon Toose's work, for example, on how granules form, or also on how synaptic vesicles form, I think that was at the time, probably together with other groups in the US, leading research. But again, as I said, I was not satisfied by that.
Yeah. I mean, I think one thing that if I think about that time that I can only, reflect on by reading papers, I mean, it looks to me that there was a great combination because it was not only cell biology with all the imaging methods and the electron microscopy, but it was the mixture with also biochemistry and all the biochemical methods that was developed, and the ability to culture cells that really, made strong advances
No question about it. Absolutely true.
Yeah, so I think again is a testament to interdisciplinarity at the EMBL and the ability to, talk to very different people with very different background to come up with solution to.
Yes, absolutely true. I mean, to give you one example, when we worked on sorting to granules, one experiment that was influenced by an experiment that another person at EMBL, Brian Burke, had done, was to make a monoclonal antibody against the chromogran proteins, and then make sure that the mRNA coding for this antibody is produced by the cell that makes the chromogranin. And then an antibody will be secreted by these cells together with the chromogranin. But since the antibody will bind to the chromogranin, the question now will be, will it perturb the sorting, or is the sorting to the granule the dominant process and will drag the antibody, which normally would go out the constitutive pathway, would it be diverted to the granule? And the latter was the case. So we proved by that experiment, which was influenced by EMBL really, that going to the granule is the dominant pathway.
All right. So I mean, in reflecting on your career and the experience that you had at the EMBL, what would you suggest to a young student that starts the career now, looking back at your experiences?
One is, do not increase your group size too fast. The danger is that you become a group leader somewhere else and at the university, and they expect you to write grants to get more money. And in the end, you are sitting in an office. You no longer do bench work yourself if you are an experimental researcher, but you give the discovery making to your PhD students and postdocs. And I'm preaching this. I tell every of the young group leaders, stay at the bench, make the discoveries yourself, unless if you are super lucky, like I was, with PhD students like Christoph Niers or Patrick Beuerle, then you can give the discovery making to such super good students. But that's the exception. Normally, your students are good, but maybe not as good as the group leader. Your postdocs, especially in the starting phase, are usually never as good as the group leader. So to let them to ask them to make the discoveries is a lost case. You have to make the discoveries yourself. And if you think of famous Nobel Prizes, like Hartmut Michel for crystallization of a membrane protein, or Neha and Zachman for patch clamp, they did these experiments themselves.
So tell me about what you think about dissemination of science. I mean, scientists are doing experiments, but the society always asks that the experiments are to be communicated. So what's your view, and how did you see this changing through the years? Again, starting back then, when...
I think the researchers or scientists have realized that there is a need to increase communication with the public. And I am actually giving a lot of public talks. So, for example, when we have in Dresden the Lange Nacht der Wissenschaften, Long Night of Science, I always give a talk to the public about our research. I think it is very, very, very important to do that. Also to explain to people that gene technology is nothing bad, but something very good. To explain to people that animal experiments are not only important, they are a must. And in fact, I am going very far in this respect. I'm telling people, and I get a lot of head shaking at the beginning. I said, listen, do you guys in the audience seriously think that a mouse model will cure a human disease? We have to make, I'm sorry to say, primate models, monkey models of human diseases. And in China, that's what's happening, right? With the rhesus colony in Kunming, as far as I know, they make a lot of animal models, and that will lead to therapies, not with mice. But I think, and in the German Primate Center, where one of my postdocs is now a group leader, I think there are good tendencies now to use, for example, common marmosets that we also worked on to make disease models. It's very important if you really, I'm a medical doctor, right? So if you really want to solve terrible diseases, you need to test them in an animal system as close as possible to us. We obviously cannot, it's illegal, and it's correct so that it's illegal. We cannot work with the great apes, that's obvious. But the rhesus monkey or the common marmoset are legal animal models, and I personally think they should be used with all the ethical standards that need to be fulfilled, of course, it's completely clear. But that's in my opinion, the best way of proceeding with curing terrible human diseases.
So, and how do you see the role of other model system? I mean, do you think that then there is a value in having the plurality of model systems and getting a synthetic view for that, or you would rather go for the more relevant things only?
No, clearly the big breakthrough, or one big breakthrough, is the organoid technology that also Jürgen Knoblich in Vienna, for example, developed, or the guys in Japan. That is very, very helpful, and it can reduce the number of animal experiments you need to do. But in the end, you need to have a disease model in a full organism with blood vessels, with lymphatic vessels, with everything, an organism that undergoes aging and so on to really get at the root of a disease.
So I mean, thinking about all the people that you met at the EMBL, we discussed about Kai Simons, Marino, and the others. I mean, what would be the best advice that you got from some of these people about your career?
So obviously Kai was and is a major factor and figure and friend in my life, that's clear. The other person that gave me very interesting advice was John Toos, the late John Toos, because we did experiments together, and when he then came with his cigar to the lab, and he had this way of very directly stating things. And he said, "Wieland, you're stupid. How can someone who is so good in biochemistry like you sit in your office and not work at the bench?" I will never forget this. And he was right.